Okay, this time it's all about product rule, linked genes, and crossing over. I just want to give you a little overview of the product rule because we've been talking in the last two videos about the crossing with one gene, then two genes, and the question, it was begging the question, what do you do with like 1,000 genes, or all of the genes of the genomes in human beings, which is more than 100,000, so more than that. Well, you can use the beautiful product rule. So as you can see here, uh, on that right side, when you look at the different Punnett squares that I produced for you. Let me just switch that over. Okay. So those are just one cross at a time with one gene at a time. Here, R and R, Y and Y and T and T. Well, only one gene. Those are after homologous chromosome have been segregated and sister chromatids separated in meiosis. So now you have only one copy of that gene in each gamete, but each of those gametes contains all the copies of all of the gene, one copy of each gene in the genome, right? We only choose to represent here only one gene at a time because it's easier than having thousands of letters, right? And when you break things down like that, you can really easily answer questions such as this one. This one can be like, okay, when you cross two individuals and you have the individual one being R, R, Y, Y, and here I'm only copying the top, all the column headers that are gonna give me the genotype of that individual, and I'm crossing that individual with the other one, which is also uh, heterozygous. So when I cross those, and it could be any type of genotype. I could give you like a 6-gene genotype. You just have to make 6 Punnett squares, right? And here we have 3 genes, R, Y, and T. We make a 3 Punnett square crossing those 2 individuals. And then I'm asking you the question, how many of, what's the proportion of offsprings? What's the proportion of offsprings that would be little r, big Y, and big T, little t? Just random. I just came up with that on the spot. Well, what do? I look at the Punnett squares, and here only 25% of offspring will be little r. So the answer will be 0.25 times big Y, only 25%, so one quarter again. So let's use the fractions. 1/4 times 1/4 again. And here, big T, little t. So here it's 2 squares, so it's 50%, so it's 1/2, right? And so the answer to that question, what's the proportion here? It's 1/4, 1 times 1/4 times 1/2. So 4 times 4, 16 times 2, 32. 1 times 1 times 1, 1. So it's 1 out of 32. So the proportion of offsprings that will end up with that genotype is 1 out of 32. I did that in less time it took me than to draw the giant Punnett squares with 3 genes, right? And you can see how fast it goes because you only have to do the little squares. And the little squares are very easy. And when you start thinking better in terms of squares, you can know right away that if you cross the heterozygous with heterozygous, the chance of being homozygous recessive is 25%. Because you can see all those squares when it's cross heterozygous with heterozygous, they all look the same. When you get used to it, you get used to it fast. And when you have homozygous, it's all the same. When you have homozygous, heterozygous, it's also a typical square. So really it gets easier the more you do those crossings. So that's the product rule. Theoretically, at the exam, I can give you that top row here and that question here, and you can give me the proportion, right? You can do that. And that those two individuals can have up to 6 genes, and you can still give me the answer really easily by making 6 little Punnett squares. Okay, let's take a. Okay, now linked genes. I want to start with that slide here. That's the last of the set of the linked genes, but I just want to show you, see if we understand what's happening here. That slide here shows us a chromosomes. So you have a chromosome here, and the centromeres can be here, whatever, right? And then different location for different genes. And so one is here, one is there, then you have another one over here, another one over there, and finally we have the last one over there. It doesn't really matter what they're called. They must be different genes, right? So it's going to be different letters that you can find at each of those loci a different gene, so different allele. Okay, now when you have on that chromosome like that, you know right away that the ones that are further apart are less linked to each other. The ones that are really close to each other are really linked, and we know the linkage about of those genes is really important about when it comes to talk about crossing over. So crossing over, if I show you the whole thing from the slide, it's when we are in prophase, right? Or even metaphase, but it's mostly in prophase. One, during meiosis, that those homologous chromosomes, they start condensing, right? And they next to each other, they get ready to migrate, the nuclear envelope is disappearing, the spindle fibers are forming, that's prophase one. The chromosome are ready to migrate to the center of the cell in between the two centrosomes to be pulled apart during anaphase 1, right? But during prophase, when they condense, it's possible that the legs of the chromosome get overlapping. And you can see that if you overlap another homologous chromosome, the same as this one with the same genes, maybe different alleles, you can swap that part with the part of the other chromosome, which is what happens here in the homologous chromosome part in meiosis 1. And that meiosis 1 here is metaphase and anaphase pulls those homologous chromosomes apart. But they steal each other's a little piece of each other's. And when we say linked, is because those two, when there's a crossing over, they are going to be carried over to the other chromosome. It's going to stay together. Whereas those two, they're going to be separated by the crossing over. That's really the important part in the linkage of those genes. And finally, how do we detect that? Well, when we look at a crossing, and the example is given to you with the Drosophila here, when we look at a normal crossing between an homozygous recessive and an heterozygous, we see what we should expect to see in terms of phenotype. The colors is handy to tell you how the phenotype should be. What you expect will be a 1-to-1 to 1-to-1 ratio. So as many offspring of that type as of that type as of that type as of that type. And when you see something different, like as we see here in that cross 5-1-1-5, in terms of genotypic ratios, we realize that it means a crossing over must have taken place because our Punnett square is showing us what theoretically we should have based on the phenotype and genotype of those parents, but we don't end up with that. That means that a part of a chromosome from an homologous pair has been spread, like shared with the other homologous chromosome, and exchanged, which changed that link. So the A and B here must be far apart on the chromosome. A might be here, B might be here, but certainly not A and B here. Because if it was the case, the crossing over will unlikely break them apart. It's still possible, but the crossing over happens with that piece only. It's unlikely, right? So that's the whole part of linked gene and crossing over, how those things interact with each other. Because we know what we should expect if there would be no crossing over, because when you make a Punnett square, you don't assume any crossing over, you assume genes are just independently assorted, right? But when they're not, this is because the genes were close to each other, right? And that's it. That's it for linked gene and crossing over. See you next time.